since we were very happy with this pilot collaboration, we expanded this effort in larger scale sequencing data. Most of these work were done by two postdocs in my lab, Dr. Morita and also Dr. Wang, who performed the bioinformatic part. So far, we have finished about 70 AML patients being sequenced with this Tapestri platform. The initial AML panel, which targets about 19 recurrently mutated genes in AML, and also which targets the 40 amplicons of these 19 genes. And samples were either sequenced by the MiSeq 150 or 250 parent end. For the FLT3 ITD cases, we prefer to use this 250p parent end because it will cover most of the FLT3 ITD. And we use the GATK haplotype caller or ITDC combination to call the mutations. And we also did a concurrent bulk NGS as well as ddPCR to validate the single cell DNA-seq detected variants. And we are very happy to see that 100% of the mutation that was called by the bulk-seq was also called by the single cell DNA-seq platform. So there's a really good concordance between the bulk-seq and the single cell seq. And we did some SNP array data, which I'm not going to show today for the time, but we also did a SNP array to confirm the zygosity of some of these cases mutation, and which resulted in this heat map, which consists of about 600 cells, AML cells from these 70 AML patient samples. And each partition is a patient, but within that patient, there are thousands of cells smashed and creates cellular level co-occurrence and mutual exclusive landscape. We also started to analyze the longitudinal samples, and we accumulated about 10 or 20 cases now. And this case really represents this longitudinal power of longitudinal analysis. This was a 74-year-old male with therapy-related AML presented with initially the 62% blast in the bone marrow. And this patient was treated with azacitidine and sorafenib, which is a FLT3 inhibitor, and achieved complete remission and enjoyed that for about 9 months. But eventually relapsed with 75% blast 9 months later. And we had a sample at pre-treatment and the relapse being sequenced by the Tapestri. And at the time of pre-treatment, this sample had a 4 independent clone, which was the FLT3 ITD, FLT3 D835E, and FLT3 D835Y, which is very tiny here, and KRAS G13D mutation. And although this sorafenib and azacitidine made this patient into remission for 9 months, the eventual relapse clone was full of this FLT3 D835Y clone. And it really made sense because Catherine Smith at UCSF previously created this kind of a nomogram for FLT3 inhibitor and its sensitivity to differential FLT3 mutations. And FLT3 D835Y clone was shown to be resistant to sorafenib, while D835E clone or ITD clone was shown to be sensitive. So it really made sense with this case that sorafenib were able to take care of this ITD clone and D835E clone. However, this very tiny D835Y clone that existed at the time of pretreatment survived this treatment and constituted the relapse. So by analyzing these longitudinal samples by single cell sequencing really created this deterministic view of how each clone independently adapted to the therapeutic pressure of sorafenib and azacitidine.